It's okay. So there's three types of mycorrhiza. So there's arbuscular mycorrhiza, which are the most primitive, let's say. Then there is the ectomycorrhiza, and then there is another one that is ericoid mycorrhiza. And so just to give you an idea, so you have a little tree here, which is about 10 centimeters, and you have like a very large network of fungi that you see here. This is the white AZ networks right below this in the ground here. They sustain a very large amount of the plant biosphere, let's say. Here you have a very nice study that is from 2 years ago, and you see the different types of mycorrhiza and the amount of plant biosphere that they sustain. So you see that arbuscular mycorrhiza are mostly centered on the equatorial forest, and ectomycorrhiza are more in the, let's say, temperate to cold biomes. And then you have ericoid, which is really ericoid mycorrhiza, which is really specialized for very cold climates. And then you have the non-mycorrhizal plants that represent only a fraction of the forest. So basically, the take-home message of this slide is to say that I mean, most of the plants are sustained by these large networks of fungal filaments. Why is so? Because the fungi provide nutrients, key nutrients for the plants. And in exchange, the plants give a bit of their carbon that they assimilated from photosynthesis. So it is estimated, it's quite hard to quantify this flux of carbon to the fungal network, but let's say it's between 5 and 15%. Sometimes people are saying up to a third of the carbon assimilated during photosynthesis is transferred, but this is really, it depends on the two partners, basically. So this is pictures from Jonathan Leake from University of Sheffield, and really you see that here in this on the right panel, you see that the radioactivity. So they labeled CO2 gas with C14, and you see that the radioactivity is really well centered after certain times of like 24 hours, I think. You see that the radioactivity is concentrated to the growing part of this system. And so basically this network filaments, they are really able to interact with the minerals way more efficiently than the root of these plants. So they are really, the interface between the plant and the soil is really mostly fungi. So I like this wording from Jonathan Leake. He's really the master of this mycorrhiza. They are really networks of power and influence, because basically the conditions, the plants, they really can access minerals that are inaccessible to the plants because of their really really large ratio of surface to volume. They are really much more favorable, these very fine filaments, to go in the porosity of the soil, detect the interesting minerals and exploit them, or organic matter also that they help to degrade. So they are really important for the plants. They transfer these nutrients from plant to plants also, that has been shown in forest. They're also able to, they are kind of a reserve of water for the vegetation, so they are really, really important for the plants. Some studies shown that there are forests that were linked to this mycorrhiza. They are able to sustain, they are more resistant to drought, for example, and they are quicker to recover from drought periods as well. So they really, in many aspects, they are really important for the whole biosphere, basically. So in my work, I really specialized in this, in the later aspect about mineral weathering. because these fungi, they are well equipped to access nutrients in quite difficult setting, let's say in minerals that are inaccessible to exudate from root plants. The fungi can really go in and do the job for the plant. And this relationship is super important for the since the beginning of this coevolution between plants and fungi. So some part of my work has been dedicated to really understand how this process of mineral weathering could be accelerated by the fungi. So here you see really two nice pictures. One from an ex-colleague of mine, Gadze. And this is an AFM view of an hyphae, and you see on a mineral, and you see that you have the hyphae and you have like this bio layer all around. And what I've been doing is really looking at this interface between the minerals here and this hyphae. And here you see on the top right, you see so this is a slab of granite that has been colonized by a network of hyphae. So you see the density of this hyphae is really phenomenal, and they really exudate these EPS layers that really, let's say, condition the microenvironment all around this hyphae. So I've been doing a lot of experiment using these microcosms. So you see, we reproduce really crudely this symbiosis. So you have a little baby tree here, and all the A's that you see, this is orange around the network of the roots is the fungi. You see here these networks, and at the level of the mineral surface, you will see way more hyphae. So what we've done, we expose these mineral rocks to this hyphae, and we let them react with this. And then later on, we used fib to cut at different sections, different position along a single hyphae. We preleve different foil of biotides, fungi sections. And then we did all kind of analysis, TEM, STEM, EDM, EDX, and also STIXM to look at the speciation of iron. I'm not going to go through all this, but some bits I'll show you today. So this is the idea. This is very simple. So you have a single hyphae here, highlighted in red, and then we cut different sections. Here you see the detail of the fibbing. I guess most of you are maybe familiar to this, so I'm not going to go too much to detail. And then what we do is profiling, chemical profiling through the interface of the hyphae and the biotite. You see a detail here of this interface, and what we see, so we usually work with silicon, oxygen, and potassium, but we do measurements of iron, magnesium, and aluminium as well. So what is interesting is really this interface here, where we see that there is somehow a delay into the rise of the concentration of the potassium here. So this interface here is somehow some kind of an altered layer of biotite that is growing due to the presence of the fungi. So if we zoom in into this section here, we could see that there is, with time, so the different section 1 to 4 is increasing biotite hyphae contact time. So you see that you have a depletion of this potassium. So this is expected now because biotite is, because of the structure of biotite, this is the potassium is located into this interfoliar space. So this is the, let's say, easiest element to move away from the mineral and move into the biological system. So no big surprise here. What is also most, interesting as well is that we see also movement from iron, and we have also data showing the same kind of profile for aluminium and magnesium. The alteration is very shallow. We are talking about, at most 40, 50 nanometers in depth. But this is cumulated over the whole length of the hyphae, which can be quite substantial. It can reach up to a millimeter, sometimes more. It depends on the way the fungi grow, whether it's exploratory growth or with this, or like more exploitative growth that we can see around minerals like phosphorus, apatite, rich rocks. So we can, I mean, there is a model that we created based on a few solid state diffusion equations. You can model the flux of the elements of interest from the mineral into the hyphae, and we can see that this is quite substantial. But the main question that we had when we saw these results was, okay, is it maybe due to an acidification processes? Because we all know that the mineral dissolution rate is really heavily pH dependent. So, you know, maybe it was due to the heavily acidified microenvironment. So we set out to measure pH near this hyphae into this biolayer that I shown you before. So we did quite a lot of confocal microscopy with a pH sensitive probe called SNAF. This pH sensitive probe is really made for measuring pH within cell, but also outside. So whether, you know, you can use special conjugated probe in order to prevent the probe to enter the microorganism. So that's what we did, and we measure the outside of this the pH outside of the hyphae. And what we saw was quite interesting. We saw that when the hyphae was in contact with mineral, with biotite in this case, we had pH which was very low. We could not measure the pH with this probe, in fact. And when it was not in contact with the surface of the biotite, we could see that the pH was around 6.5, which is somehow expected for this microorganism. So this was quite surprising. And to date, I mean, I've done this measurement in 2011, I think. There was, I could not measure the pH of this hyphae. So that's still a question that is still not answered. So I would be interesting to measure that today with new probes that can go to lower pH. But basically what we show with this is that the fungi is really able to acidify this microenvironment. And that's maybe the explanation why we see an increase of the rate of dissolution at this interface. When we calculate the rate of dissolution of this mycorrhiza based on the flux of elements that we measured at the interface and the pH that we measured near this hyphae, we could define this region as the region where the fungi was dissolving the mineral. So if you compare to the bulk literature, you may think, okay, that's not really fast. But then you have to consider the environment on which these fungi are doing this. They are sitting basically on the basal plane of this biotite. And this basal plane has very, very low reactivity compared to the edge. And most of this dissolution here, this is dominated by the edge dissolution rate. So you have to deconvolute basically the two rates between the basal plane reactivity and the edge plane. And when you do that, you realize that the basal plane has a very low reactivity. And this is, let's say, 3 to 5 times lower than what the fungi is doing on this basal plane. So on the basal plane, the fungi is way more active in terms of weathering than the same pH reactivity of the basal plane. So this was quite a surprise to us. We could not really figure out why there was such a high reactivity on the basal plane of this mineral. in presence of this hyphae. And one observation that we did, which was also very surprising, is to look at this reacted biotite surface after the colonization of this hyphae. So you see here a few pictures of SEM of this biotite surface, and you see that there is what I call trenches that are formed by the presence of the hyphae. This was not present before. And what you saw here, what we saw here is also that there was some kind of a pulsatile behavior. So we know that the fungi is growing by pulse. So you grow, you have a turgescence that is really building up at the tip of the hyphae, and then it grows in one go, and then it repeats this growth. And on the biotite, you see these pulses. It lets some traces here, and yet there is material that is accumulated at the end of the pulses. So, and we know that biotite, it's kind of an easy, it's not very tough material. So if you humidify it a bit, and you can really easily stretch it and create these trenches. I mean, people have been doing AFM on this reacted surface, and they saw that the depth of these trenches is about 100 nanometers. It depends on the trenches, but it's quite substantially deep compared to the surface. So it's really like as if the hyphae was really scratching, creating more surfaces, creating more edge surfaces. So I believe that after colonization, this whole surface of the biotite must be way more reactive than what it was initially. So this is an observation that was really surprising. And beneath this, when we look at the FIB sections as well, beneath the hyphae, we saw that there was also misorientation of the crystals, and didn't show this data here, but we did electron diffraction in this area and beneath, and we saw that the crystals were not aligned. They are not really in the same direction. So there is definitely some kind of a forcing of the crystalline structure of the biotite. And we saw also that there was a lot of cracks that are formed, and we saw that in many different FIB sections. When we compare that with the control FIB section, we didn't see that amount of cracks. I mean, this was really localized below the hyphae. So that's still something that was hard to understand at the time. But then we measured this the speciation of iron, and we realized that the speciation of iron was changing as well. We could see that there was some oxidation of the iron 2 in the biotite into iron 3. So this was something that was quite unexpected. We could not really understand what was the process to oxidize this iron 2 into iron 3. So, but this is really something that we measured on at least one ETH. So we did the same as we did before. We took different FIB sections and then exposed that to STIXM, and we saw that there was a huge difference between the tip of the hyphae, which was not so much oxidized, and the iron 2 iron 3 ratio was similar to what we could find in unreacted biotite, and the FIB section that we could sampled a bit further upstream the hyphae, where the iron 3 to iron 2 ratio was way more oxidized. So that's still something we don't understand. We don't know why there is this oxidation. Usually fungi would reduce the iron in order to assimilate it. So that's still something we're still figuring out. What was the process at work here? But what is important is that if you oxidize a certain amount of iron into biotite, some of this iron 3 that you are oxidizing is going to form subdomain of iron 3. And we believe that this iron 3 subdomain within the biotite that we could see sometimes here We could see regions where the this spacing was similar to fire rate, for example. We think that this is creating a strain um, in the structure of the biotite and create these cracks. So that's our hypothesis at the moment. Um, but we could see that there is all kind of change in the mineralogy, in contact with the fungi. So here you see the interface between the fungi here on the right and the biotite. So usually the biotite, when it's pristine, the despacing is around 10 nanometers. But then here you could see regions where you have 14 angstrom. Sorry, this was 10 angstrom before the pristine biotite. So we could see that there was region of 14 angstrom despacing. So which is similar to what you could find in vermiculite, for example. We could see also these different cracks in the structures. So this is really there's something happening with this biotite when it's reacted with the fungi, and we think this is related to the oxidation processes. So this is, just to show you that, I mean, this is just a cherry picking in many different aspects of this fungal mineral weathering. I could have talked about biotite weathering, where we see also a huge increase of the weathering rate with this mineral. So what is important to understand is that the fungi really accelerate the mineral weathering and for the plants, and it does that in environment that are really hostile for the plant. For example, where you have very shallow soil or very low amount of organic matter in a soil, the fungi really predominates the flux of nutrients from the mineral soil basically into the plants. So fungi are really helping the plants in tough conditions, let's say. And this is really, if you think of the transition from, aqueous, the life from aqueous system into the land, this would be the ideal partner in doing this transition. The plants would really benefit from this partnership with fungi. So here is a little sketch that I have done for that summarize a bit the action of the fungi for the plant. So the plant gives some carbon to the to ensure the growth of the hyphae when it needs nutrients. The fungi will grow. Then there would be some biomechanical forcing, channeling. There would be some acidification processes, sometimes oxidation of the mineral, especially in the case of biotites. And then you would create, a layer of altered material that will tend to grow, but at some point the flux of elements, I model that the flux of elements would decrease really substantially, and therefore there would be an incentive for the hyphae to grow and to contact new surfaces. So there would be some kind of a feedback process, let's say, that ensure that the fungi will continue to grow on the surface, at least of the biotite, and you renew the flux of nutrients from the mineral into the hyphae, and therefore you have the cycle that starts again. So I think there is, this whole process has been studied in laboratory mostly, and in very limited set of minerals. But I think there is an interest to expand this research. Recently, I've seen that there was an interesting study by Damien Daval done in the eastern France, in the Strainbar catchment, where they really measure, at least they try to measure the weathering rates of olivine in natural conditions, and they found that 16% of this weathering rate was assigned to fungi. So I think it's quite substantial. That's deserving, more research, I think, because I think olivine is a specific cases as well. So it's a mineral which contains a lot of iron. So the iron would tend to somehow block the weathering, I think, for fungi. So I think this is, this was the research that I was doing when I was postdoc and early into my position here at ULB. And then I expand my views into deep times. So I got interested into fossil by interacting with people that work in Congo. We are in Belgium, so Congo is somehow related to Belgium for good and also bad reasons. But what we have here is an unprecedented record of Congolese geology in the Tervuren museum. So I got access to this core, and I started to play with these samples. And I started to understand a bit better the association between plants, early plants, and these fungi. And I got interested into the Neoproterozoic times, because this is, and I show you better later, this is really the period where everything happens for this the creation of this partnership between the plants and the fungi. So what we know is that the early plants, they have the ability to interact with an unprecedented diversity of fungi. So you see here the liverworts, they are this, they are very minute plants. You can find them in very humid environments. If you go to a shadowy spring, the spray of water will humidify the soil or the surface of a rock. And if you look closely, sometimes you may find this kind of little plants. They look nothing, but they are super important to me, I think. So they are part of this group, this Mycorrhizal, and people in Sheffield and also in Leeds have been analyzing this the ability of this plant to bind to different types of fungi, primitive fungi. So you have this Mycorrhizae, you have Glomeromycotina, the Basidiomycete and the Ascomycete, they are more evolved fungi. But you see that this plant can talk, let's say, with this fungi. They can create mycorrhiza association with this fungi. And this is really, really telling a lot. I think these plants, they were really the key into the colonization of lands. And this is something that has not been reproduced in later plants, much later plants. Only the conifers can really achieve this kind of feats. So when you look at these liverworts, they are really primitive. They are dating back to the really early plants like Ordovicians, Silurian. They were there already. So I got interested into the early records of fossil of fungi. And when you talk about records of fungi in geological times, you are talking about the Rhynie chert. This is the most well-known rocks for studying fungi fossils. And what is striking in these rocks is that you find already quite a large diversity. If you look at the different groups of fungi, this is a very simplified phylogenetic trees of the of fungi. So Ascomycota and Basidiomycota, they are really the most evolved. They were called the higher fungi. And then you have Glomeromycotina, Zygomycotina, and Chytridiomycotina. These groups, they have been changing all the time. So I simplify here. So they are the the lower fungi basically. And in Rhynie chert, basically you find all four of these: Ascomycotina, Glomeromycotina, Zygomycotina, and Chytridiomycotina. So only Basidiomycotina is absent. So that means that at around 400 million years, there were already a large, the diversity of fungi were already there. So that means that this diversity must have been evolving way earlier in history. So that's where things the things are starting to get really complicated, because when you look at the fungi records back in time, in Precambrian times, this is really messy. Well, you have in 2005 you have a group from China that discovered filaments. I don't know if you see my cursor here, but basically these filaments here, they claim it to be fungal, of fungal nature, and you have this this kind of algae, these cocoids, that are present there. So they are interpreting these pictures as an early lichen-like fossil, and it's dating back to 600 million years. So we have also other type of enigmatic fungi, assimilated fossil, putative fungi. We have Tapania from Nick Butterfield. So basically, it's kind of a potato shape fossil with kind of ornament. So this looks like filaments. So this was interpreted as the early fungi. And then really later on in 2017, there was claims of a fungi which was much older, like 2.4 billion years old. So this structure here, so they are chloriterized network of filaments. So it's really hard to, there was no traces of organic matter at all. But when you look at the morphology of these networks, it really resembles really strongly to fungi. You see anastomos, so fusion of two different filaments. So this is really something that only fungi can do. So this was interpreted as a fungi living deep into the ocean, deep in the ocean floor, into basaltic vesicles. So this is really specialized. This was really a weird setting for fungi, because this is most probably anoxygenic. So this really shook a lot of people that were interested into fungal records. And then in 2019, there is another fossil of fungi that has been discovered from in the Arctic, from the Neoproterozoic, around 900 to a billion years old. And what they did is also, so most of these studies are mostly, they are studying the shape of this fungi, and really rarely they were looking at the quality of the organic matter and the structure of the organic matter, and trying to identify this organic matter and relate that to a fungal fossil, let's say. This study did that in a way. It's, you can criticize that, but at least they tried their best. So they did FTIR measurements on this filament, and they found what they think is chitin. So when I looked at that, and I thought maybe we can do better than that, and that's where I studied this core in in the Museum of Tervuren, because some of my colleagues here in Brussels, Alain Préat and his PhD student Frank Del Pomdor, they attract my attention to fungal. I mean, what was claimed fungi at the time in 1973. There was a paleontologist from India that claimed to have found fungal network into a core that was contained in the African Museum. So we looked at fib sections that we've done on this core, and we found some of this filament. So this is a picture that was taken from this paper in the 70s. So you see these filaments, and what was interesting is that later specialists in the 90s, they look at these pictures and they say, well, okay, this is not fungi, this is cyanobacteria and tubular of cyanobacteria, and that's it. And discussion was closed. And I thought, okay, maybe we could have a look at this fungal network again and see whether this is cyanobacteria or not. And that's what we are doing. I'm thinking that I'm running late at the moment, so I will try to go faster.
No, that's okay. I mean, we
Yeah.
have time still. Yeah.
So this is a log of the core. I'm not going to go too much into detail, but this is mostly, we found the fungi in, well, the fossil fungi in a dolomitic shale level here in what is called a B2C 8. And the paleo environment of this section here, it's really sub aerial. It's a kind of a perennial lagoon. Sometimes that has been drought. We see that in the different FIP section, not FIP section, but in the different fin sections that we've done. Paleontologists and also sedimentologists have interpreted this paleo environment to be really close to the terrestrial domain. And when we look at these new fin sections, we could see this kind of structures. They are quite large. I mean, these networks of filaments, they are about 500 by 500 micron. And when we look at this in optical microscopy, we could see this is black, non-translucid, and you see this. There is like this branching here. This is natural color here. You see these black filaments here. They are branching here, and there are multiple branching, several other branching. The length is quite large of these filaments, and when you look at the width, it's really around, the average is about 6 microns. So this is what you can expect from fungal network. So this is more pictures, SEM pictures here, and the interior is hollow sometimes, and this is cylindrical. So we assign this cylindrical preservation to the fact that this is dolomitized. So the process of, usually the filaments and all, fossils, they are flattened because of the compaction. In this case, this is not this is not what happened because of this dolomitization. The fungal network stayed as in 3D. So this is quite exceptional. And the big question was whether or not this was fungal. Based on the shape, you can have an argument, but this is really not very strong. So the whole thing was whether or not we could identify chitin into this fossil. We know that chitin can be preserved very well in the geological records. It has been found in arthropods, but also in demo sponge from almost 500 million years old. So, this is something that can be preserved really well. So we set out to do this. But first I did the classical, characterization for organic matter when you want to look at fossil. We did Raman, and so we identified the typical D and G bands at 16 and 1350. And we started to play with this spectra. We did some mapping. We measured also the peak temperature of this organic matter. You can derive that using a nice ABAC. I think this is somebody from Paris who did that, Besac. And you could derive the peak temperature that the organic matter has been experienced. So for our fossil, this is 150 to 250 degrees. And in this range of temperature, you can preserve chitin. It's not going to be dissolved. And what is interesting is that I did this ratio between these two peaks, so this G band and the D band, the D1 here, in order to derive the structural order of these organic remains. So the structural order, this ratio, I'm gonna use that in conjunction with a ratio that has been used many times in fossil research. This ratio is the methyl to methylene peak that you can find in this aliphatic region of the FTIR spectra. You see that you have a very large peak here of the aliphatic region. So you have this two peak here, these two peak at 2960 and 2920. You can use that to derive the degree of branching of the organic matter that you are looking at. I'm not going to go into detail here of the whole spectra here. What is important is what we found here in this region, the fingerprint region at 1540 and 1650. This is the amide 1 and amide 2 peaks. And so this was really giving us a lot of hopes, because this is really what you need to see in order to identify chitin, because chitin is an acetyl glucosamine compound. So you need to find an amine or an amide group into this fossil in order to claim the presence of chitin. And we also find a small peak of a pyranose, so it's a leftover of a sugar or carbohydrate. So this is, the two pieces here are important. This strengthens our claim of the presence of chitin. But what is important is that I use this ratio of this CH3 and CH2 peaks in order with this, in conjunction with this structural order index that I use, that I derive from the Moraman, in order to separate, well, let's say to separate whether to determine whether this fungi was, this fossil was coming from a prokaryotic organic matter or prokaryotic, organism, or eukaryotic organisms. And so this is quite a new way, I think, to separate these two groups. And this is, so I use different data from Q and et al. From 2015, 2018 in order to separate these two groups. And for our fungi fossil, we fell really well into the region where we expect the eukaryotic fossil to fall. So this is really a nice fit here for this fossil. We could show that this is an eukaryotic origin for this fossil. So I'm not going to spend too much time on explaining xanes. I guess everybody's familiar to this. What is important here in this slide is, so we did a fib section here in this on this network. We placed that onto xanes, and what we see is three main peaks. Here, this is the orange spectra that you need to see. So we see an aromatic peak, very prominent. This is normal because of the kerogenization of this fossil. But we see also a shoulder here in this region, which is what you would expect for the amide carboxylic region. And we see also a peak here into ketone and aliphatic region at 286. So basically what, how you would interpret that is that this peak that you would find into the chitin has been degraded into these two peaks here. So the aliphatic, this aromatic peak is due to the kerogenization of the fossil, and then you have a phenolic peak here that is really coming also from the degradation of this organic matter. But what's important is that we still see a shoulder here into this amide peak. So I'll show you quickly the same xanes, but this is on the N edge here. And we see also we have an amidal peak here for the nitrogen. So this is consistent. I mean, this is not diagnostic of the presence of chitin, but this is still a strong indication that this is consistent with the signature of chitin being present into this fossil. But what really nailed the presence of the chitin into this fungi fossil was the presence of the labeling that we've done with confocal microscopy, fluorescence microscopy. So we use a specific dye for chitin, and then we expose some regions of this filament to this dye, and then we did confocal microscopy on it. So you see different pictures here. So some are completely dark, some are whitish. So the whitish show you the presence of the positive presence of chitin, and the dark here show you the natural autofluorescence. So organic matter can have a natural autofluorescence, so you need to check for false positive signature. So in this case, we see that the dark here, this is the organic matter that we've taken from a different slice of the core, of the Kanchi core. So we see that the organic matter, the old organic matter, doesn't react with the labeling, so there is no big problem. So this is natural autofluorescence. So this is the fluorescence that we measure when the labeling was present. So the organic matter doesn't react with this labeling. So it seems that this labeling is really going to target the chitin, even if the organic matter has been matured. So there is no false positive, basically. That's what I'm going to say here. But the interesting thing is that the network that I've shown you before on ACM, so we recognize it now. We see this branching here, and what we see is also that we have this cylindrical signature here. So this is really something that indicate chitin being preserved, colocalized with this with the membrane of these filaments. And we saw also some septum. So this was also a bit surprising, because septum in a fungi is synonym of quite higher fungi, so evolved fungi. So if we look at this really ancient fungi, it's quite a surprise. But in fact, when the septum are very, very infrequent. We see a few septum, and sometimes they are not complete. So, in other words, they are not obstructing the whole cell. And this is really what you expect when you look at higher fungi. This is really a barrier between the different cells of the fungi. In this case, this was really not the case. We could see that this is incomplete septum, and this is quite rare to see them into this network. So this is really what you would find in a very primitive group of fungi, which is called Blastocladiomycota. So we think, this is really an hypothesis here, that this is this kind of group that we are seeing here in this fossil. So this is my last slide. So what I've shown you here is that fungi are important nowadays because they sustain a lot of the major forest and major plant biomass on Earth, but they were really important into one of the most important transition of life from liquid to land surface. And I think fungi were there, and they were really helping the first organisms to step on the land. And what is really striking is that this connection probably started earlier than we think. It started probably already into this little ponds or this lagoon that we looked in in this Congo core, because there was a really interesting studies from the Fries that show that some group of green algae, they have basically the genes, the genome packaging, if you like, to talk to the fungi. So I think this association started, in these ponds that are subjected to drought periods. So this fungi and green algae, they interact physically to each other. Maybe they were parasites at some point. Who knows? And that's the right environment to create this symbiosis. And some people have been trying this. To associate green algae with fungi, and it worked. After a couple of generations, they were forming a free living association. So I think this is really the kind of environment that are really essential for this transition. And I think what we have in the Congo core is maybe one of this environment. In this core, there have been a lot of green algae, architage of all kinds that have been identified. So far, we haven't seen this connection between these filaments and the algae, but maybe it's just a question of looking really for this kind of weird structures. But that's all. I think what I have to say. I think I have no slide anymore. Yes, some people to thank at ULB, the GaveZ, Lian Benning, and also some people from the Carnegie Institutes, people at Diamond as well, who helped me a lot with the Xanes, and of course the museum here in Brussels.
Okay. Well, thank you so much, Steve. And it was a very fascinating journey through fungi world, I will admit. We're gonna take a few questions. So I know Francois has raised his hand, but anyone else have a question in the meantime while Francois is asking question, either you can raise your hand or wave at me, type in the chat, either one. So Francois, you can start.
Thank you, Steve. Merci, Steve, for l'excellent exposé. Thank you very much. Yeah, I was very interested by this older work, oxidation phenomena of the biotite by the fungi. One question, of course, these fungi, they have a metabolism at this moment, which is oxygenic respiration, right? Is it correct? And can it be quantified or in some
Yes,
way?
I think it can be quantified. I never tried, but yeah, it could be quantified. Yes.
Yeah, because it's quite fashionable these days to invoke once you have a kind of oxygenic respiration, you have a reactive oxygen species moving all around.
Yeah.
And it's really fashionable in geochemical cycles now that it could really account for a few percent, maybe several tens of percent of the oxygen flux. And in that case, of course, it would be interesting to relate the oxidation of biotite to generation of reactive oxygen species. And I was wondering, because you showed very nice pH probe images. I know there are probes for the reactive oxygen species, and there are kits and things like that. So I was wondering whether there was research done in that direction. And
I like to do that. I tried, but yeah, Colin Hanser.
Yeah, for the manganese.
Yeah, for the manganese, she used. I don't remember the name of the compound, but I saw the picture where you see the hyphae and you have this blue color that showed that there was superoxide O2 minus present. So that's something I really would like to do. I tried here in Belgium to convince the FNIs to give me money to do that, but they were not really keen.
Okay, but yeah, that seems to be very interesting, and that could be in the trajectory of oxygen radicals and things like that, from the photosynthetic thing to the fungi. That's a really exciting field, and I think
there is a lot of people here are working on, for example, lacase for degradation of organic matter in soil, in wood, for example. And yeah, that's something I would love to do, but you know,
Okay.
this experiment with fungi, they are really killers, and I was really happy to have people like Jonathan Leake to really give me a hand on that. And so far, I've been trying here in Brussels to set up this, you know, this thing to create again these microcosms. It's, you know, it's tough.
Yeah,
And I've been unlucky with, we started that and then COVID came, so all died. So, you know.
Oh.
En tout cas, merci beaucoup, c'était extrêmement intéressant.
You're welcome.